Hi guys! So in this video we will talk about the central dogma of biology that explains the flow of genetic information from DNA to mRNA. And this process is called transcription. So what you see on this side are the components of transcription that I'm going to talk about in this video. What you also see on your screen is this really scary looking picture that will eventually make sense to you when I'm talking about these components of transcription. So before I start with initiation, a couple of notes. A lot of people get confused between terms coding strand and template strand. So let's get that straight first. We have two strands on the screen, 5' to 3' and 3' to 5'. And one of them is a coding strand, one of them is a template strand. It's very simple. The coding strand is the strand that has the promoter sequence embedded in it. So this strand that has the -35 consensus sequence and -10 consensus sequence is the strand that has the promoter sequence. Simple. This is our coding strand. And this, the strand that is complementary to our coding strand, is our template strand. How do I know that this is a -35 and -10 consensus sequences or the promoter sequences? You just have to memorize the nucleotides involved in a promoter. These two sequences together make the promoter sequence or make the promoter. So this is my promoter. Apart from determining the coding and template strand, the promoter also determines, especially the TATA box, the -10 consensus sequence, also determines the transcription start site, which means the first nucleotide that will be transcribed is also determined by the promoter. Usually, the transcription start site is about 8 to 10 nucleotides downstream of the -10 consensus sequence. So 8 to 10 nucleotides from this consensus sequence, let's say up around here, would be our transcription start site. This is where the actual mRNA would be transcribed. This would be our real product of the transcription. The mRNA would start here. So initiation starts with the sigma factor. So this is my sigma factor that binds to an enzyme called RNA polymerase. So this is my RNA polymerase. And together they bind to my promoter sequence. So they bind to this promoter sequence here. So let me just erase this real quick. So this is my promoter sequence and it goes and binds to the promoter sequence. So let's do this. My RNA polymerase and my sigma factor goes and binds to the promoter sequence. So this is my RNA polymerase plus my sigma factor. Binds to the promoter sequence, and it creates something called a closed complex. After it's bind to the promoter, it has created a closed complex. What it starts doing, the RNA polymerase plus sigma factor, is that it starts breaking the hydrogen bonds between thymine, adenine, and cytosine and guanine, converting the closed complex into an open complex. One important thing about the sigma factor is that without the sigma factor, RNA polymerase cannot bind to the promoter. So this alone cannot bind to the promoter. RNA polymerase, if it cannot bind to the promoter, then it cannot synthesize a new mRNA strand. Sigma factor is really, really important. This whole thing forms the initiation complex with the promoter region, sigma factor plus RNA polymerase bound to the DNA template strand. So this is our initiation complex. Our next step is the elongation. This RNA polymerase starts here after it proceeds through to the transcription start site, starts here, and after transcribing about 15 nucleotides, a change must occur to the RNA polymerase. The sigma factor must come off. So my new RNA polymerase on this side should look something like this without the sigma factor. If it needs to transcribe beyond 15 nucleotides, this sigma factor should come right off. RNA polymerase should not need the sigma factor after transcribing about 15 nucleotides. Elongation constitute of adding ribonucleotides to the strand, to the 3' hydroxyl ends of the previous ribonucleotides. If I have A, C, G, T on my DNA template strand, my mRNA would start with U, G, C, A. This would be my mRNA, and this is a completely hypothetical example. So, after my RNA polymerase synthesizes my mRNA, what then happens is that my RNA polymerase goes on to synthesize something called a terminator sequence that would help terminate the transcription. So once my RNA polymerase synthesizes the mRNA, so this is my finished mRNA, what it does is it transcribes these. These are the terminator sequences. There are two types of terminator sequences in transcription: row dependent and row independent. So first we'll talk about the row independent termination. What happens is that we have something called inverted repeats. These are inverted repeats. What they do is that they form a hairpin like this, which is a very destabilizing structure, followed by a string of uracils that are complementary to these adenines. This is a very destabilizing structure, and what it does is that it destabilizes this complex, this RNA DNA hybrid. It destabilizes it, and what happens is that RNA stalls and then falls off, cleaving this mRNA in process. And this is row independent. So this is my row independent transcription. Another kind of transcription is my row dependent transcription. So what happens in row dependent transcription is that we have something like this. We have a 5' to 3' and 3' to 5' DNA strand, and this is our mRNA that's being synthesized from 5' to 3'. Remember, the synthesis in mRNA is also always from 5' to 3'. An enzyme called Rho is attached to the newly synthesized mRNA strand. What it does, and this is my RNA polymerase here synthesizing everything. What it does is that when this RNA polymerase encounters this termination sequence, let's say this is my termination sequence. When RNA polymerase encounters this, it stops, it pauses for a while, and this Rho protein catches up to this RNA polymerase, and after the encounter between the Rho and RNA polymerase falls off and cleaves this mRNA. And transcription is terminated. So we have row dependent or row independent. And that is my terminator sequence right here, the inverted repeats. And the product is my mRNA. And that's it.